fantastic. I presume you're all going meeting people, but because I can't see, I don't know what you're all doing really. Could all be on holiday. But I mean, the idea here is that biotech, TechGate, digital is connecting loads and loads of people, but it's not got that feeling of a conference, because the missing part is, if you like, the gatherings. And this is an example of one of the gatherings. We call it Coffee Buddies. Graham and I have done nearly 130 of these now since COVID started. We've done so many. It's just quite amazing, really.
I had a full head of hair when I started.
And it's been a fantastic journey, and it continues to be a fantastic journey, because I think we all see that the future is going to be the hybrid model of meeting each other, but also carrying on this. And the festival that we've got this week is fantastic. We're doing, how many are we doing, Graham? 16, or is it more?
16 over the
16 of these interdispersed at 9 in the morning, 12, 3, and then 6. So we pick up all the time zones. And we're also lucky enough to be having the launch of the first international XY competition for really companies that are looking for visibility. And the idea, we call it XY because we don't want to just call it the X factor, because somebody's got that. So it's the XY factor. And it's really looking at the secret sauce of what is it that makes a company special. And we're lucky to have MSD that are involved in this, and we've got some other investors and some other people who are experienced. And it's a competition where we're not sure what the prize is, but it's something to do with spending time with Graham. But we've got this competition, which is 2 events, and we're really excited because we're getting some interesting companies turning up. And one of the things we've realized from this doing this before is how you never know who you're going to bump into, and that's what this is all about. You never know, your dream customer could just be one coffee away. And
can I just say something as well? We've got a couple here, Amy Lamb and Alyssa came and said they met on Biotech Gate last year, and they're doing business together. So there it is, evidence right there.
Yeah, so I mean, this is what it's all about. So, and to catalyze our conversations, we're lucky enough to have various folks come to talk to us. We would have a conversation, and then we're going to move into the moving everyone around and getting to know you. But we're lucky tonight to have Stina, who's turned up to talk to us about what she's up to. And Stina, welcome. It's a real delight always to see Stina, and tonight's an exceptional delight, really. Delight to see Stina. And Stina, who are you? What do you do?
Well, thank you, Tony. Yeah, I'm Stina Lundgren, calling in from Sweden. So hi to all of you. Nice to see such a crowd spread out through the world, and always a pleasure to join this Coffee Buddies event, I think. I don't know where to start, who I am. I'm a medicinal chemist. That's my kind of background. But I've been working with drug discovery for the past 14 years, and the last 2 years with Pelago Bioscience, focusing on target engagement primarily.
Fantastic. And the world of small molecules is moving along. It's making leaps and bounds, you know. So this idea of target and engagement, these are 2 words I'm interested in. So maybe you could explain to the folks what you think of as a target, and then maybe what it is that is an engagement.
Yeah, I think a target, of course, is what you want your molecule, which could be a small or a large molecule, to interact with. In one way, perturb, you know, affect, change the status of that. We are mainly focusing on protein targets, but you know, a lot of people are starting to look at RNA as targets, for example. But if you would say a protein target, and engaging that is then to affect the functional state of the protein, and that could either be that you have engaged the whole pathway, so pathway engagement, or engaging a specific target in that pathway that in turn has a functional effect and phenotypical effect.
So, I mean, there's some interesting language here. So you know, in the days when I've been doing these things, you get a pipette and you put it on a cell and something happens. So what can you see that I couldn't see when I put my pipette and dropped something on a cell, and it, like a dendritic cell, and it went wild? What do you see that I would have seen with my going wild, but is more detailed?
So what we study is the effect on either globally in all proteins in the cell, which are affected. So kind of translating the phenotypic event you're seeing by looking what's happening globally at all proteins. Or it could also be a specific protein that we study more closely. And I know either you're working in a phenotypic-based drug discovery where you don't know your target, or a target-based drug discovery where you actually know your target. And to me, it was always, you know, you start off with something, a recombinant protein in a biochemical assay or a biophysical assay, and then you move into the cell assay, as you just described, and you were just confused.
Yeah.
But then adding this piece of data of looking at the target engagement will make you less confused, at least, and can, it's like this compass that kind of turn, and you suddenly see where north is, and you can navigate yourself towards the direction you
read an interesting paper to get today. It fascinated me. It described things called guilty targets. I love that terminology, that there are guilty targets. You know, there are targets that are particularly the ones you go after. And so maybe could you say something a little bit about this technology that you've got, where it came from, and what it is about?
Yeah, sure. So it's an invention that's from Per Nordlund's lab at Karolinska. So that's where it came from originally. And then based on the invention and this technology, it was spun out a company then called Pelago Bioscience. And the technology is in essence quite simple. So it's treating either cells or tissue with the compound, and then applying a short heat shock of 3 minutes, and looking at then after that, reading the effect on the thermal stability of the proteins.
And I mean, where is it starting to have an effect? You know, where in the drug development or drug discovery world is this sort of this technology giving a solution to something?
We're doing a lot of projects for customers in early lead generation, so applying it already to identify new chemical matters. So that's definitely one area where we see a lot of value by doing this quite early. But then I think also for confirming your hits, so later when you actually have some chemical matters, to confirm that target engagement in cell is another application that is widely being used.
And if you, I mean, you can look at tissue, can you
Yeah.
not? So tell us something about how you can look at tissue. I mean, can you take biopsy of a synovial joint, or take a biopsy of a knee, and you take a bit of synovium, and can you ask the question, is my drug there and is it binding?
So we wouldn't answer if the compound is there, but we would answer the question whether the target is affected by something. So we're not studying the concentration of the compound, but the effect on the target. And just as you say, that could be done in tissue. And then later, I mean, in the preclinical drug discovery pipeline, that's where you see this technology adding additional data by actually looking at in vivo samples, for example. But also you can have quite rare patient samples to look at the target engagement in that quite early, and then correlating that with a dose, for example.
And I mean, how do you think this is going to change the way that we discover drugs? Are people going to build different sort of screening cascades, Stina, where, you know, there's a...
I think it already has changed, because we see CETSA being used already quite commonly early on for hit confirmation. But the real value, I think, is to bring that assay further on in when you move your product forward, as you say, in translational studies, But also what we started to look into is how it can be used on patient material. So in a clinical study, how can this support the preclinical activities in studying the target engagement in patient sample. So
what would you imagine these are going to be blood samples? Are these going to be biopsies? What are you going to possibly work with CROs who are collecting it? What's the sort of the vibe there?
I guess this depends on the of course on the disease, so the indication, what makes sense in terms of which biosample. But blood, for example, I think that would be quite good if it's relevant to this disease, to draw blood, heat it, and then look at the melt of the protein. And we're doing a research project at the moment where we're studying the effect on Keytruda, on PD-1. And the initial results is, I mean, of course, we see effect on the PD-1 and the thermostability, but it's also quite a large spread on the donor-to-donor variability, which to me is quite interesting to see if that could be helped to guide also really early on which patient would benefit most from having the treatment.
So just tell us a little bit more. So what you're saying, that if you take 3 donors, they look different. Is that
they
what
look different? Yeah, we all are different, and we are all affected differently by the compound, which is not surprisingly. But how can we use that data? That's what's be really cool.
I mean, traditionally we would approach it with a blunt object, really, you know, like a drug that hits a guilty target with a hammer, and you know, because we're all different, but we're all the same in certain ways.
Yeah,
we all take oxygen in, so there are common, but then there's this, it's subtle really, isn't it, Stina? Is it a subtle difference, or?
Yeah, I would say it's a subtle difference in most cases, and I think that actually taking a sample before you have even treated the patient, and then being able to see how that proteins will react with the compound would be quite cool.
So fantastic. And I mean, is there any people that you really want to engage with or talk to more? You know, who's the sort of group that you really want to get through to, and can we help you find those people?
Yeah, I mean, first of all, of course, anyone that is interesting, I'm happy to engage with and tell you more. But then I think actually this application of using technology at later stages, I think we have just started some initial projects internally, and I think there's a huge potential for this.
And that is somebody who's got samples of what sort of sample? You know, if I was in university, what would you want from me?
Yeah, if you would have samples and then also link it with a response, that would be really interesting, of course.
So if I was doing a small clinical study where I understood, you know, what's been going on, and I'd got consent to store some tissue, that's the sort of person that you would like to. And is there any therapeutic area that is, or is this agnostic of therapeutic area?
Well, it's an agnostic technology, so it's not dependent on the indication.
Fantastic. So if there was a couple of things that the folks should take away from this conversation, what would you like them to take away? What's the tech takeaways?
Yeah, in the importance of target engagement, and then the features of the technology, that it's label free. You don't have to fiddle around with labeling your compound. You don't have to label your protein. It's just call us and we're ready to go. And
really, you look to solve problems, so you're delighted to talk to people when they're in the problem solving area. Really, you know,
just call us and tell us what your problem is.
Yeah, and then you can help. So I think fantastic. Graham, shall we just see if there's any immediate questions in the group here? Graham, you're on mute.
Trust me to be on mute, but yeah, if there's anybody with any questions, if you'd just like to put your hand up, if you've got any.
Hang on,
Peter, if you take yourself off mute
Peter,
and ask your question, that would be great.
Hi, Stina. That was very interesting. Thank you. I've been involved in a project for the last 6 years called VisiCourt, and the purpose of which is to determine why certain people reject transplants of the cornea.
Mm-hmm.
You know, there's no real metabolism in the eye, so it's always a mystery why something so small can cause such a reaction when it's transplanted. Well, we have a vast biobank of samples collected over the 6 years, which are designed for, I suppose, research in ophthalmology. So if you can think of potential applications, it could be an interesting one to look at.
Oh, thank you. Yeah.
Yeah, plenty of proteins.
Yeah. We love those.
Yeah, there's a lot of protein in the biobank. Other questions, otherwise I might pick on someone.
Tisha.
Hang on, we've got
Tisha's
Tisha.
got a question. I thought you would, because it sounds like your company is generating lots of compounds. So go ahead.
Tusha.
Yeah, thanks, Stina, for a wonderful explanation. I was wondering how high throughput, or what is the throughput of these technologies?
It depends on which format we're talking about. So the protein wide, when we look at around 6 to 7 proteins in one experiment, that's very low throughput, but a lot of data on different proteins, of course. But then we have the plate-based method, focusing on a single protein target, and that has a much higher throughput.
Okay. And just one more question. So how do you discriminate between, target and pathway, for example, changes?
Yeah, so what we usually do is to run a lysate experiment side by side with an intact cell experiment. And in the lysate, we would see the direct binding proteins being affected, and the biology is off. And then compare that with the intact cell, where we will also see the associated proteins being affected. So, and depending on the time point, of course, we would see more of the phenotypic response of the cell, but keeping the time short, we would mainly see associated proteins being affected, like phosphorylated proteins and such.
And can you quantify this effect? For example, if 2 different drugs, they are showing same effect, but can you quantify between them?
Yeah, so we can also do experiments where we vary the concentration of the compound, but keep the temperature fixed. And then we would get an EC50 of the compounds, which it would then be a way to rank them.
It would be. Have you ever done any immunohistochemistry? So have you ever taken? Because what would be fantastic is to stimulate a cell, get them to light up like a dendritic cell, so they're hugely lit up because they're, hugely stimulated, and then ask the question, what proteins are, in your words, perturbed? Because I love that word. Have you ever done that? Have you ever done? Could you do it in a section, do you think, Stina? Can you take a cell out of it? Can you actually do it in situ?
Well, we've done that in the targeted format, where, not we, but Karolinska, our collaborators there, they looked at that, but that was on a specific target. I don't know how we could do it in the protein wide, but definitely it's possible in when you focus in on a single target. And then you also can get the information on in which components the protein is being engaged.
Yeah, no, a fascinating technology.
We're getting to that time where we think about
just one more question. Maybe we could have one more question.
There's one more question before we break out. No, I think we're good to break out.
We're good to break out because it's 26 now. So let's move on. Graham's going to, Stina, firstly, can I just thank Fab in case we lose anyone in the journey, but Fab, thanks very much. We'll give you a round of applause, but we'll give you another round of applause later. But that was terrific. And it is fantastic technology. It's absolutely extraordinary. I mean, from an empirical systems biologist such as myself, it's just fabulous to see what drugs really do. I just love it. But Graham, over to you for this.
Yeah, I'm going to create 3 breakout rooms, and what we're going to do is 15 minutes in each. We're going to do 2 rounds. So it's amazing how quickly 15 minutes goes. You know, quick introductions to the people that are in the room with you, and yeah, just garner as much information from each other as you can. We're going to have lots of more breakout sessions throughout this event, so you know, there's
I was thinking before, because we don't hand out business cards, if you want to put your name, you know, in the chat room or to share it, that's a good idea.
We do have it. Yeah, that's right. Everybody's names anyway, and I think it's you could look on the partnering system, couldn't you, if you wanted to meet up and connect with each other during the event. So anyway, I'm going to, but do whatever you want to do, of course, to get your name out there. Open up those rooms now, and here we go. The algorithm's working. If you just accept the room that you're in, and we'll see you back in 15 minutes.